right, let's go ahead and get started. So happy last lecture. So I guess you've officially survived the 200 series, so that's good. And then, of course, just a reminder, so we'll say that any physical copies of things, so right, homework, labs, and then ideally the final, those are all due to the mailbox Friday, so that'd be tomorrow at 5 p.m., so June 6th. And then anything else, so electronic assignments, you have until next week to finish up. So if you still have homeworks, labs that you owe me, right, you can email me copies of those or submit them on Blackboard. And then same thing with the paper. So the paper you can email by June 13th, or maybe I'll make a Blackboard turn-in. I haven't quite decided. If I make a Blackboard turn-in, I'll make an announcement, but email always works as well. And anything else that's straggling, and again, don't forget those reading questions. Some of you have neglected them, so use that last week. Hopefully, again, you're kind of free from my final the last week, so use that to finish up reading questions and everything for completion. Questions, comment, last minute things, and then obviously poster presentations today. So that will be exciting during lab. And I'll kind of go over the format more, but basically you'll at least try to see, like, I don't know, 4 posters each round or so. I'm gonna have to print out more mini rubrics for y'all. And so you'll just kind of grade each other loosely, and then I'll come around. So of course make sure that I see your poster, and then your peer scores will probably be like averaged or something and added to your overall score. But again, I expect people to do well, nothing to get nervous about. We're just practicing our mini presentation skills. Other questions, comments?
Where was the mailbox again?
Oh, so my mailbox is just on the 3rd floor. It's on like the windows side, but if you get lost, you can ask the secretary. But just go on the far side where the windows are, and then you should see all these boxes, and then yeah, you'll find my name. Other questions, comments? All right, so let's look back at a few things. So I think your book, sorry, I have too many windows as usual. So your book kind of does, is this the correct chapter? No. I was going to say your book does what I call like a poor job on the whole DNA thing, honestly. So I kind of wanted to go back over that and at least make sure we hit a few key points. So your book, right, they just kind of talk about what they're coated with and who they pair with and the hydrogen bonding, so that's great. But let's make a few things just a little clearer. And so it kind of depends too, like what we want to talk about. So obviously DNA replication is a pretty big topic. And so I guess I want to clarify that first, because it's like, are we talking about DNA replication or are we talking about protein replication? And I just think we should be a little clear about that. And so we're looking at this chart here. And so again, this is telling us how to code for different amino acids, which obviously make a protein. And so let's start with that. So let's look at how our DNA essentially codes for proteins. And then I also will post a bunch of links to other videos in the chat. So especially too is like you move on to OChem, there's like Mastering Organic Chemistry guy and a bunch of like OChem tutors that are really good videos. And so like I said, I'll post them for some of these DNA ones because like I said, there's a lot of different processes. Your book doesn't do it justice at all. We're probably not going to do it justice today, but I at least want to try to hit a few highlights and again go over this protein thing first. But there's a lot of great resources out there as well. And so first of all, let's be a little more clear about our DNA ladder structure. And so here we're given this kind of picture in the book, but your book doesn't give a good depiction of how it's actually set up. Oh, and now I lost my... window. I have too many things. Okay, so basically I was trying to pull up a picture, and so you kind of have, maybe you see this cartoon representation of, okay, we have our 5' to 3' strand, and then the complementary strand is 3' to 5'. Well, let's first back up and what the heck does that mean? And so that has to do with the direction of these groups. And so basically one strand runs in the 5' direction, so based upon where the phosphate group is and how it's synthesized. And then this one, if you kind of look at the phosphate group and where it's starting, so this is in the opposite direction. So that's why this is called 5' to 3'. And then, right, the complementary strand runs opposite of 5' to 3'. And so that might explain to the answer key. So we'll look at your homework as well in the answer key, because to me it's written kind of confusingly when it just the way the answer is, because it's rewritten in the 5' to 3' direction. All right, so now we have that. And then, like I said, I apologize for having to use my personal computer. I'm like too much of a ADD person, so I have like way too many tabs and I always lose everything. All right, so then let's start with that. So, okay, we have our 5' to 3' strand, 3' to 5'. And so, okay, for protein coding, so again, obviously, right, we all have our DNA that tells us how to make stuff. And so then our DNA can be transcribed to mRNA. And then, right, this goes off into the ribosome to make whatever protein we desire. And so, of course, what kicks off this process is our RNA polymerase. And so I just wanted to show some of these structures. And so this is a structure, sorry, like I said, trying to pull over the stuff I pulled up for us. So this is a structure of our RNA polymerase. So this was actually a big deal. Let me pull up the paper actually. So this is kind of cool. I wanted to show you all this in case we have some people that are more into proteins. And so this is back in 2016 where they were visualizing this RNA polymerase. And then what was really cool, I mean, the paper's admittedly really dense, but they try to show it in the different stages of transcription. And what's also really cool, so if we go back to this structure here, so here's our structure, and then it tells us the technique that they use. So like I said, I'm mainly familiar with like crystallography, and that gets down to a certain resolution. In this paper, they use a special electron microscopy that's like specially made to get down to a pretty small resolution, so 7.9 angstroms. So again, I would have to kind of look at how that compares to an X-ray crystal structure. And it also depends on the quality of each data collection. So that's what kind of these resolutions are for, so you can, again, appreciate how they got that. So with electron microscopy, again, they're able to give our whole structure of our polymerase. So that's pretty cool. And now I've lost the Nature tab. And so I just want to pull up their paper. And then I don't know if y'all have a sense at all of like calibers of journal articles, but obviously Nature is a pretty high caliber. Technically, this goes into like things that are only relevant if you're going to grad school, but each paper has like an impact factor, like each journal, I mean, and so sometimes PIs make goals to get a certain impact factor. But anyways, Nature is very high impact. And then I just wanted to show one of their figures, if I can find it. So obviously, right, they're taking a whole bunch of pictures in all these different stages, which was pretty cool. So I'm looking for, 5C, because this was cool because, again, they showed it in the various stages of transcription. So that's cool. Figure 5C here. Where did it say what? I was looking in the paper. Oh, here it was. That they're looking at them disconnecting the DNA strands at 5C. So that's kind of cool. Again, just to kind of see how that type of research would go. All right, so then, like we were saying, so our RNA polymerase comes along. And so this also, just like an FYI, can be inherited right initially by mom. So that's how baby has some initial RNA polymerase, right, to start synthesizing all the proteins. Because, right, that's kind of the question of, well, how do you synthesize proteins without this polymerase, right? Well, you had to inherit it. All right. And then, so it unzips it. And then the bottom strand is considered the template. And so, of course, then this goes on to provide a template, right, for protein making. And then if we remember, so kind of going back to some of these notes here. So looking, just reminding ourselves that we have uracil when we are in RNA mode. And so, right, A pairs with T. Unless we're making RNA, then we have to pair A with U there. So just keep that in mind, make a note for yourself. And so let's look at some of the homework. If I can now find it. All right, sorry, so trying to find, let's look at, we'll look at these two. But I'll provide some criticism and talk. All right, so let's skip 5 for now because I don't want to talk about DNA replication quite yet. So we're just focusing on proteins at the moment. And so then it asks us in 6, what is a complementary mRNA strand of this DNA? So see if you can do that, but then we'll look at kind of right how the answer key reports it, what are they trying to tell us, so that just so we're all on the same page there. So those are, sorry, clicking the wrong notes. Yeah, so those are bases, right? So these are just bases. And then they're going to code for the amino acids. So sorry, I'll go back to the, like I said, let's just look at 6. And so then we could start just kind of sketching it out, right? And so, again, we're kind of taking a little bit of an oversimplified approach, perhaps, in this class. And so we can just write our complementary, right? 5 prime, so G with C, A with T or otherwise U since it's mRNA, and then G with C, C with G, and you guys can help me make sure I don't mess up, T with A, C with G, C with G, didn't give myself enough room, and then A with U again for our mRNA, so 3 prime. And then what's confusing to me is, right, your answer key writes it in the 5 prime to 3 prime direction. So all they've done is report it, right, as CUC, so right on the key, they start 5 prime CUC and just write it in that direction, G, A, G, G, U there. And then I don't know, this example kind of bothers me from your homework just because every 3, right, should code for a protein. And so let's look at that now. And so again, we've done a poor job as chemists just trying to introduce this of actually coding for proteins. And then this is for our 5' to 3' direction, so we'd look here. And so usually, this is just kind of an aside, right? There's some sort of like a promoter region that starts it off for these proteins, and it's usually called the Tata box. So those of you in biology may be more familiar with that. And again, feel free to step in and correct me, or if you have any comments. And then there's usually also some sort of tail end, the poly A tail, which just protects it from any sort of other enzymes or possible, degradation in the environment. And so then to kind of read this, so obviously there's a lot of different combinations to get to the same amino acid. I kind of like the compound chem chart better for that, just because you can see all the combinations that lead to a specific amino acid. But of course, right, like there won't be the opposite, right? So basically GTT is always valine, right? There's no other possible amino acid, but there's 4 different ways, right, to code for that amino acid. And so then we could, do some practice of, right, just writing out different codes. So if we had U, obviously that would be for, I think that's phenyl. I don't know my amino acids. I never formally took biochem, so I wasn't forced to memorize them. So we can look up phenyl over here to remind ourselves. So yeah, phenylalanine. And then, so let's look at some other things. And so then going back to DNA, so then what I kind of don't like is I feel like we almost confounded the 2 processes. So right, it just asks like, oh, what's our complementary DNA strain? But then we don't really get into DNA replication at all. So that's like a much more involved process. That being said, you should totally watch some videos on it that I'll post. But let's at least do like a quick and dirty, I'll just say very quick DNA replication, just to kind of compare. So right, the processes are a little different. So obviously the proteins go off and are made in the ribosome. And then DNA replication is semi-conservative, so meaning, right, we're going to keep half of our helixes. And so essentially, right, we have our, man, I don't know if I have these numbers correct, but we have, right, our 5' to 3' in one direction, and then 3' to 5' on the other. And then they unzip, and then of course we can make, right, our complementary strands on both sides. And so this is where we probably want to watch some videos on this, because like I said, the DNA replication is a little more complicated of a mechanism because one of them is synthesized in just the forward direction of the 5' to 3', while the other one has a special, I think it's an enzyme that kind of goes piece by piece backwards and kind of has to go around. But like I said, that's probably a little more involved. So if you take biochemistry, obviously you'll get into all the DNA replication in more detail. But then we could at least kind of practice, right, what they want us to get from here. And so of course, if we're just talking about DNA and DNA, we're not going to have U, so that's all that we're trying to get here. So try to practice that. And like I said, we'll look at how the answer key reports it. And so let's go over this. So again, right, we want to sketch it out. So this is going 5' to 3', then this is opposite, right, of 3' to 5'. And so G was C, A now with T, right, because we're with DNA, that's really the only distinction. And then G was C again, C with G, T with A. I didn't give myself enough space. And then CC, so G, G, T, 3'. And then once again, right, your answer key reports it in the 5' direction. So starting 5', so C, CTC, GAG, GT. So that's, again, just something to watch out for. So if you report it, right, 3' to 5' like this, I'm not going to take off either, right? I understand that. But just, it is helpful to put this 3' there. And again, you want to understand that one strand runs in the 5' to 3' direction, and the other runs opposite.
So, if you're writing the answer key in the 5' to 3' direction, are you still matching each base with the one adjacent to the 5' from that direction? So
yeah, so it matches, right? So it starts, so that's why I sketched it out here to here, right? But then I'm just telling you that the answer key reports it in that direction, and then that's generally also like how they're synthesized. But like I said, we're kind of taking, right, a simplified approach, because like I was telling you, the DNA runs in two ways, right? So one of them runs in just a normal direction, and the other does a weird backwards 5' to 3'. But as long as you, like I said, match them, and then I would denote which end is a 5' and which end is a 3'. Otherwise, it does get confusing. And then, so other kind of things to look at that kind of came up, or things to think about. So this is kind of cool. So this is, again, just from my chemical magazines that I get a billion of. And so one of their latest stories was about protein folding. So we kind of mentioned on Tuesday, right, that some of our diseases come from secondary misfolding, so in the hydrogen bonding. Oh, and with that, sorry, I have too many things going on. There's also this, maybe this is why I don't like biology, because you have to pull up like a million pictures to help. And so we were just looking at the standard codon table. So this was obviously in your notes, but something that I wanted to just point out, and this goes back to like our secondary structures and how it starts to fold, is some people prefer this wheel. And so it's based on position 2. And all that means is position 2 here tends to kind of predict how hydrophobic or hydrophilic the amino acid is. And so that's all they've done, is arranged it in this pretty chart so we can just kind of see the amino acids. And so you can, of course, start to think about how that might affect folding, right? So if these are in an aqueous environment, obviously there's water and hydrogen bonding. So if you have something hydrophobic, right, it's going to kind of turn inward and turn away from the water, right, and try to fold inward to the protein. Whether something is hydrophilic, right, it's going to turn outward. Yeah.
You mentioned using the letter U as a base earlier.
Yeah, so that's for RNA.
RNA.
Yeah, so that's only in RNA. So that's what I was saying. We kind of went over those processes a little quickly. So if we're talking about DNA to making proteins, right, it goes DNA to RNA to protein, and then DNA to DNA just makes itself. So, right, you preserve half of the latter there.
Does the U base replace any letter in particular in the...
Yeah, so it replaces A. So you don't have A in the RNA. But yeah, so going back to this, again, if it's hydrophilic, it might turn out. And so a lot of research has been done over the years, especially recently. I don't know if you've heard of like those protein folding things where you can, apparently donate your computational power to protein folding. And so part of that, or part of the culmination of that research is kind of shown here and what they're trying to do now. Obviously, everybody likes to use this AI buzzword, but what they're trying to do is play with exactly that, of potentially like how could you change these secondary interactions with their hydrophilicity or phobic nature. And so that's all they're trying to do here. So that's kind of cool. And I mean, they describe it as like an arm waving. I think that's just a good way to try to get people excited about proteins and boring things. Like, yeah, we're going to have these little motor machines waving or whatever. I mean, I think it's still cool just because of how they're trying to manipulate that. So again, obviously, right, that's future research, and obviously pretty difficult too, right? But trying to figure out how could you maybe design this protein a little differently? How can you influence the folding? Maybe you could even think about how do you cure some of those diseases that we talked about? Could you refold it somehow? Who knows, but kind of interesting to think about there. All right, I wanted to touch upon the sugars a little bit more as well, just so you can start prepping yourself for OCHEM. And so there's kind of two things that we should talk about. So first of all, right, we have these sugars, and so we're looking at them in this form, but there's actually a process, and so this goes back to our different projections. And so there's this thing called tautomerization, if I can spell it, or tautomerism, or the process of tautomerization, where it just basically means we can do some electron pushing, but for sugars, this means that we can go from a ring structure to an open chain, which is a pretty low percentage. So this is probably like 0.02%. And then it can flip forms. So this is kind of beyond our scope. But this is called the Pyranose form. And then it can flip and form the furanose, which is the 5-membered ring. And so this is 6-membered. And so let's kind of look at that, because this is where you get all this interchangeable. So I didn't draw. So let me draw that out. And so what I mean by that is I had mentioned perhaps earlier when we were looking, actually let me pull that up again if I can. Oh, let's look at Wikipedia since now I've lost my glucose. So let's look at glucose again. And so we looked at glucose, right? And then you look at these pictures and it's given to you in kind of both these forms and then also this Fisher projection. So now this is explaining why the heck it comes in all these different forms. And it even mentions, right, pyranose, so that's kind of hinting at, again, the 6-membered ring form of glucose. And so that's something that you want to, again, get familiar with. And so let's look at glucose again. And so now let's figure out how to convert between these two. So again, you won't have to do this for this class, but it's good to start thinking about this to prepare yourself for OCHEM. So we want to be able to go from our Fisher to a Hayworth. And the Hayworth is just the ring looking thing. And Fisher is going to be our flat one. And so Fisher, sorry, I want our glucose for reference, so you can look at Wikipedia, look at this picture, and then we'll renumber it and feel better about it, hopefully. Oops, that's not how we feel better. All right, so there's our glucose from Wikipedia. So D-glucose. And so now I'm going to write out the Fisher projection and how it's supposed to help us. And so there are these little line things. And so then we have CHO, so this is carbon 1. And then OH, HOH here, OHH, OHH, and then CH2OH, and then we can start numbering them.
Would you put an H by the first OH?
So this is, well, this is supposed to be a 1, and then this just says CHO. And then were you asking about an H here? Yeah, sorry, I skipped that. So carbon 1, 2, 3, 4, 5, 6 there. And then, so to label it, yeah.
What is that called? The like CHOOH.
The straight line here.
Yeah.
So this is just called a Fisher projection. And so I'm drawing glucose. And then so kind of, there are some tricks, they call it. So sometimes people tell you to kind of visualize this as someone trying to give you a hug, and then that helps tell you the stereochemistry. So again, kind of in your mind, so I'm just going to rewrite it. And then you can draw little wedges coming at you again, right, as if both sides were coming to hug you. So this is going to tell us, right, which one's up, which one's down is what we're trying to get at. So this is, again, I'm just trying to give you different ways to help see this. So 1, 2, 3, 4, 5, 6 there. All right, so now they're kind of coming out, so they're wedged. And so kind of the other thing to say is, like, if it's where it is, again, tells you whether it's up or down. So then looking at these, so some tricks, and these are always like, they're helpful tricks to help you figure out. So if you're looking at carbon 2, 3, and 4, then if it's on the right side, it's down. And so something else, I don't know if this helps you as well, is like you could also think about trying to like tumble this and rotate to get kind of our side view, right? So then we can just kind of tumble this. And again, we're looking at it in its open chain form. So 1, and then I'm just trying to draw out the stereochemistry so we can see. So our hydrogen's coming out, OH is coming out. And so I guess that's like one of my main points here is this is not a flat structure, right? So that's kind of the hard thing, I think, if you're initially trying to look at this sugar, is it looks flat, right? But it's really trying to tell us all this 3D information, just shorthand. So 1, 2, 3, so I'm losing my carbon count. So I'm trying to draw our tumbled. So 1, 2, 3, 4, so hydrogen up top, OH on bottom, 5, so C, OH here. All right, so all we've done, like I said, is just kind of tumble it to C. And so then we could see how it might make, right, a new ring. So we have 1, 2, 3, 4, 5. And so they could chain up and again go to our ring form. And so now let's look at our ring and compare this to all these hints that we had looked at. So again, right, a few different ways to visualize this. So we can first think about our ring closure as well, if we want to think about that mechanism. So we're going to have the electrons on our OH. It's going to attack, what do you know, our aldehyde here, and then get us in our ring form. And so now let's kind of go back to our ring form. And so now we can compare. And so looking, so we said, okay, C2, so 1, and let's label these, right? So this our oxygen closed our ring. So this is over here. And so then this is carbon 1, 2, 3, 4, 5, and 6 up here. And so then we can see, okay, carbon 2, it was on the right side, so it's down. So 3, right, it's on the left side, so it's up. 4, right side, down. And then, so sugar R5 here. So looking, again, if it's up or down, that tells us if it's D. And then our carbon 1 as well, so that tells us whether, again, it's our alpha or beta. So of course this one is down, so then our alpha D sugar there. Yeah.
What is it, show alpha or beta again?
So it's these two that we're looking at, and so this one is down. So these are both down, so that's alpha, but let's look at, so this is another picture of the alpha beta thing. So looking at, again, position 1, and then you can compare it to the one across at 4. So down, down is the alpha form, and then if it had been up, down, so opposites, that's the beta.
What if it's up?
Then it'd probably be like, because then you'd rotate it.
Oh.
All right, I think that at least kind of helps. Let's do a quick demo that I brought. So I brought our aspirin synthesis, not aspirin, I don't know what I'm saying anymore. Nylon, so nylon 6,6 synthesis. And so I'm using slightly different reagents than maybe we've seen in our typical condensation reaction. And part of that is because I want it to go. And so I am using hexamine diamine. So let's draw that out just for kicks. So 1, 2, 3, 4, 5, 6. If I can do that, 1, 2, 3, 4, 5, 6. So hexane diamine is one of the chemicals. And then instead of using a carboxylic acid or something, using something a little bit more powerful. Oh, come on. I was trying to show off the picture of it. So I'm using adipole chloride. So notice that we have a chlorine here instead of an OH, right, for our leaving group. So it's going to proceed by the same mechanism. I'll at least sketch it out. So plus adipole chloride. And so I suspect that if we had used the carboxylic acid, that it would just be a lot slower. Sorry, I can't count. 1, 2, 3, 4, 5, 6. And then chlorine. So again, right, we're going to have a pretty similar mechanism like we've seen. So nitrogen can attack here. And then, right, there's going to be proton transfers. Can you tell what's going to leave, though? So instead of water, what are we going to be forming? So you got a Cl. So that should normally be an OH. So what combines then? What are we forming in this reaction? Not NaHCl, but so remember the nitrogen doesn't go off with the water, but you got the right idea.
Hydrochloric acid.
Yeah, hydrochloric acid. So this is a little nasty, so it will fume slightly. And then once I get it all done, then I can rinse it and we can touch it. So let's, oops, pull up our demo here. Let me. All right, so let's do our reaction here. Sorry, let me remind myself of the prep. I think I remember. No, I don't remember. Alright, sorry, I'm following a lab and I just want to make sure I at least roughly put in the correct amounts. Oh, I forgot something. Hang on, I forgot sodium hydroxide. Alright, let me hand out the quiz while I go run and grab some sodium hydroxide real quick. I have to grab something. So you can at least start looking at the quiz. I guess I'll be right back with sodium hydroxide. Oh, it's not on there? Yeah, it's not on there. Yeah, that's fine. I'm sorry. Behind on everything. Oh, yeah. See it? Back here. And so we've already prepared a solution. So the hexamethylene diamine, we just, it's a solid powder thing. You have to kind of go excavating, I like to joke. But nice big solid powder. So it has now been made into a 5% solution. And so that's the first thing we're going to do, is just put, my prep calls for about 8 mils. Kind of YOLO it a little here. It's a pretty robust reaction. Alright, so we have roughly 8 mils of it. And so then I forgot my sodium hydroxide, so probably a little bit of an activator or something. Get it going. So 1, 2, 3, 4, 5, 6, 7, 8. Big measure. And so now go grab another beaker. I forgot that I was supposed to mix, so I need to mix the alkyl chloride and dilute it with a little bit of hexane. So I'm gonna go grab another beaker and I'll drip it down the side. Sorry.
Didn't want her to look at me.
So yeah, alkyl chloride is pretty nasty. Sorry. I swear, I didn't have crazy brain till this last week. And I forget everything. So I'm just diluting it. My cyclohexane, it said about 8 mils. All right, so now let's see if I can do this nicely. And so obviously we have an aqueous layer, and then I have our organic layer, cyclohexane. And so now I'm just going to drip it along the side. And you should start to see a film forming. I think it's forming already. You see that in the camera. So I'm just kind of going slowly. And so there are gases involved. It's just going to be like this, like y'all determined, a little bit of HCl. So this is looking good. So I don't know if you all can tell on camera, but there's a little bit of gas being released, so that is definitely our HCl. All right. So now if all has gone well, I should be able to grab it. And so now it kind of looks like a snot thing or something. But now if I'm careful, oops, I already dropped it. We can kind of pull it out for a while. And so I'll just kind of keep going. Oops. And I shouldn't have touched that with my hand. And so you can see, right, the nylon there. So I can come around too. So you can see it just kind of keeps going. I did not do my best job on this. There it goes. So it's kind of spidery, gross thing. And so, right, again, this could be spun into actual nylon, or obviously pantyhose or other clothes purposes. Huh?
I was just like, is that technically just a fabric?
Yeah, that's what nylon is. Literally, it's synthetic polymer. All right, so I'm going to pull this out some more and rinse it. I'll probably just go wash it in the sink to be most safe. And then I'll dry it, and we can pass it around, and you can touch it. I'll leave the stream going in case there are other questions. Let me go wash this nylon though. You can touch it, it's been washed.
You just wash your water?
Yeah, I'm just rinsing it so all like there's no acid and stuff. That's the main danger.